Hey, good evening. We'll wait for a couple of minutes to see if anybody else joins and then we can start. So, good evening, everyone. I guess we can start and let people join as we progress. I'll just share my screen. Is this visible? And am I audible?
Yes, ma'am.
Yeah, thank you. So, good evening, everyone, and welcome back to the last session in this series where we are discussing previous year assignments for the NPTEL online course Drug Delivery Principles in Engineering. So, I'll just go ahead with some of the topics that were discussed in week 12. I'm assuming you guys must have gone through the lecture material, so I'll just highlight few topics. So, we have been looking at vaccines for the past few sessions, in which in the last session we have focused a lot about cancer vaccines, and then what are prophylactic vaccines, therapeutic vaccines. Then we had discussed about what is autologous vaccine, allogenic vaccine, etc. And following up with, since the course is drug delivery, we have looked at some very specific examples. For example, there is something known as stimuli responsive drug delivery, where once you inject the patient with a drug, you want that drug to be released only at a specific only at a specific time period, and only for a specific amount of the drug should be released. So this is where we want to have control over the delivery of drugs, and we want to design a system such as whenever you inject it, we can easily switch it on or off depending upon some stimulus, some externally given stimulus. So this can be something known as magnetically controlled, where you have injected your delivery system into the drug delivery system into the patient, and then you have externally applied some magnetic field at a particular location in the body, and because of the effect of that magnetic field, the delivery system is now releasing drug. So similarly, this can also be something like ultrasound control, where you give ultrasonic waves, or it can be pH responsive. We have looked at one example of pH responsive. If you people remember, we have looked at something known as enteric coatings, where you coat your drug molecule by a polymer which will specifically release the drug into the intestine, and whenever it's given orally, it will prevent the release of this drug inside the stomach depending upon the pH of the polymer. So that is one example of pH responsive. Then there is something known as temperature responsive, or it can be light sensitive, where you shine light at a particular location. Let's say you implanted your drug delivery system in the knee of a patient. Let's say it's an osteoarthritic or arthritic patient, and your implant is now in the knee, and you want the drug to be released at specific time periods in the entire day. You don't want the drug to be continuously present in the body. So you'll shine a light, and depending upon the wavelength of the light that you have designed the system, it will then release the drug. Or it can be ligand sensitive. For example, let's say you are designing something for diabetes, and you want the drug to be released. Let's say it's an insulin drug. You want the drug to be released only after the patient has had food. So let's say you make the system sensitive to glucose. So whenever there is a high amount of glucose in the blood, which happens when you take food, that's when the system will release drugs. So these are some of the examples of stimuli responsive drug delivery. And a bit more about pH responsive and temperature responsive hydrogels is discussed in this week. pH responsive, as I said, one example is enteric coating. Another example can be whenever your drugs are uptaken by the macrophage system, it will be taken up by phagocytosis. So the drugs will land up in the endosome. You don't want that, because once it goes into endosome, then it will fuse with the lysosome, and your drug will be degraded there. So you want the drug to be released in the cytoplasm of macrophage cell line. So what you can do, you can put the drug into a polymer. The polymers are specifically chosen polymers. For example, let's say they have a lot of COOH groups, carboxylic acid groups. So whenever these polymers end up at lower pH, which will be encountered in the endosomes, where the pH will be lower than 5, so here they will become more hydrophobic, because they will be insoluble, and because they are insoluble, they'll precipitate out. That is, they'll become more hydrophobic. And since the polymer is hydrophobic, your endosomal and lysosomal membrane is also hydrophobic, so they'll try interacting with it, and in this process they'll disrupt the endosomal membrane, and all the drug will then be released into the cell cytoplasm. So this is one example of how pH responsive polymers can help. Similarly, temperature responsive hydrogels. So here we use some specific polymers known as LCST polymers. We will discuss more about this in the questions as well. So these polymers are special, meaning so if you have a normal polymer and if you heat that polymer, its solubility will increase. Now in contrast to that, LCST polymers are special because they, as you heat them up, as you increase the temperature, their solubility actually decreases. So if you make a hydrogel out of this temperature responsive polymers, these hydrogels will shrink at higher temperature while they'll swell at lower temperature. So you can modulate the polymer chains in such a way that you can increase or decrease the pore size of these polymers, and in that way you can have some control over the drug released from these polymers. Now this was about stimuli responsive drug delivery. There is another concept in drug delivery known as targeted drug delivery, where you want to specifically target a particular organ or a particular cell or a particular tissue in the patient's body. Targeted drug delivery can be divided into two parts: passive targeting and active targeting. Passive targeting is nothing but where you don't make use of any specific interaction and you just direct that drug to a particular organ. For example, we have seen EPR effect few lectures back. EPR is nothing but enhanced permeation and retention. This is seen in tumor cases. So wherever your tumor is there in a tissue, nearby the tumor it will have leaky blood vessels, because that's how the tumor is growing uncontrollably. So it does not have any organization in the blood vessels that are going towards the tumor. So since these are leaky blood vessels, whatever cargo you're delivering, let's say you have made a nanoparticle to target the tumor, so these nanoparticles will then escape from this blood vessel and enter into the tissue. So that is one example of passive targeting, where you're taking advantage of this tumor architecture to deliver your drugs. Another example can be, let's say you want to specifically deliver drugs to lungs, so you can, rather than going the oral route or injection route, you can the parenteral route, you can directly go to the inhalation route, where most of your cargo which you're delivering will end up in the lungs. So this is example of passive targeting. In contrast to that, there is something known as active targeting, where we're taking advantage of the specific interactions of, let's say, antigen antibody. You have a particular cell, you want to deliver your drug to that cell only, so it will have certain antigens presented on that particular cell. So what you can do, you can take a polymer, you can encapsulate your drug, and on the surface of this polymer you can conjugate it with an antibody which will be very specific to this antigen. So because of this antigen antibody interaction, the drug will only be released inside that particular cell. One example of active targeting for drug delivery is aptamers. This is a relatively new concept in drug delivery. Aptamers are nothing but short oligonucleotides that can fold into a unique tertiary structure. Oligonucleotides meaning these are DNA or RNA strands, more specifically nucleotides, and they're very short in their nature. So these might be some 10 to 20 nucleotides attached together. So you have a chain of these nucleotides, and because of their unique interaction, they will just fold amongst themselves and form a tertiary structure. Now this tertiary structure might bind to some proteins because they're complementary in nature. Let's say they have a specific fit to some proteins in the patient's body. So you can again take a drug, encapsulate it in a polymer, and you can conjugate this polymer surface with this aptamers. And since they will have a particular shape which will be complementary to an antigen where you want to deliver the drug, they can easily bind to this protein through hydrophobic interactions because of the shape complementarity, and then they can deliver the drugs. And then moving ahead, in the last few lectures in this week, nanotoxicity was also discussed. So we have been looking at making nanoparticles, microparticles using different polymers to deliver drugs for over 12 weeks now. So since these are foreign materials which we will be injecting into a human body, there are a lot of concerns associated with this. So first of all, there is toxicity associated with these nano and microparticles owing to their nanometer and micrometer dimensions. Because now, since you have broken down a particle into smaller particles which are nanometer in scale, they have higher surface area. So they can interact with a lot of reagents, they can interact with a lot of immune cells in the body, and in this process they might be toxic to the patient's body. So before even approving these nanoparticles for any treatment, we have to test out whether they are really toxic or not, and what is the limit at above which they can generate toxicity. So this testing can be done using several models. First is subcellular systems, where you want to see whether your nanoparticles are causing any oxidation of the proteins and the DNA present in your cells. So what you can do, you can take a test tube, you can put in normally found proteins in this which are present in the human plasma, and you can check whether, and you can put your nanoparticles in this, and you can check whether these nanoparticles are causing oxidation of this DNA or proteins. That is one way. Next, you have in vitro models where you can take macrophage cell lines, or you can take fibroblast cells and perform the same experiments in vitro. You can expose these macrophage cell lines to the nanoparticles which you have made, and then monitor their growth, see if it's toxic to this cell line. If it's found that it's okay with it, you can then go one step ahead and do toxicity analysis on isolated organs. Let's say you have taken whole blood and you want to see if these nanoparticles are causing lysis of RBCs or lysis of WBCs. So there you can test whether it's toxic to the blood. And finally, the last model to check toxicity is in vivo, where you take smaller animals like mouse or rat, and you deliver your particles to these animals, and then you again monitor their growth, you monitor their health, and see if there's any toxicity caused. So then once it's fairly seen that there is no toxicity caused in animal models, you can then move ahead towards primate model, and then ultimately, since whatever drug you're developing, whatever system you're developing is ultimately to be used into humans, so then to pass this, to get approval for treatments in humans, there are certain regulations by different regulatory bodies such as FDA, and there's a huge process before any new material that gets approval to be injected into a human body. So there are several clinical trials. You guys must have heard about clinical trials when people were trying to develop COVID vaccines. So these clinical trials are nothing but human studies, where these are divided into different phases, where phase 1 is safety assessment. You take a couple of patients, let's say this is done in collaboration with hospitals. So let's say you take a group of 10 patients and you try giving them your nanoparticle dose at a very low level to see if there is any toxicity, if there is any adverse reaction. If it's seen that there is it's completely safe, then you move towards phase 2, where you now increase the dose. This is where you want to determine what dose is okay for a human patient, and you see and you also check whether this dose is effective at a smaller scale or not. And if it's found it's okay, then you move on towards phase 3, which is a very crucial clinical trial study. This is where you get FDA approvals for your drug treatment, and this is done at a very large scale and at different places, just to see whether most of the population is okay with the drug delivery system you have developed. So these were some of the points that were discussed. So with this background, we can then move ahead with the last assignment. Does anybody have a doubt? Shall I move towards the assignment then? Am I audible? You guys can hear me, right? Okay.
Yes, ma'am.
Yeah, thank you. So let's move on towards the questions. The first question is, what are GMP and QS regulations? If anybody wants to answer, I'll wait for a while.
Number 2, ma'am.
Second
Sorry?
option, ma'am.
Can you please repeat? I think 2 people tried to answer.
Second option, ma'am.
Okay, second option. And somebody else was also trying to answer. Do you have
Yes,
this?
second option.
Yeah, okay. Yeah, thanks for answering. Yeah, both of you are correct. GMP regulations apply to things which are intended to be used in humans. GMP is nothing but good manufacturing practice. So these are certain regulations whenever you want to get approval for something that is to be used in humans, you have to follow GMP, that is good manufacturing practice. This is nothing but the regulatory bodies which are going to give the approval. They will check whether the drug which you have made is in a completely sterile condition, is made in a clean environment, there is no contamination with the drug you have made. Also, the way you have packaged it, the way you're labeling, storage, installation, everything will be checked to see if there is any contamination in the final formulation which you have made. So GMP regulations are applied to things which are intended to be used in humans. Same goes with QS. These are quality standards. Is this clear? Okay, then I'll move on towards the next question. Next question is NIPAM is a temperature responsive hydrogel. The following are its characteristics. We have to choose one right answer, which is a characteristic of temperature responsive hydrogel.
Option 2, ma'am.
Option 2 there. What is our degree? Okay, anybody else wants to try? Thanks for answering. No one? Okay, let's discuss the answer. Yeah, you are absolutely right. The right option is the second option, that is the water solubility decreases with increase in temperature. So, as I said, temperature responsive hydrogels are made with special polymers known as LCST, which is low critical solution temperature. So, these polymers actually become less soluble as you increase the temperature because they are made from both hydrophilic and hydrophobic parts. So if you guys remember, hydrogels are essentially made of hydrophilic polymers because they have a tendency to absorb water and they swell. But for temperature responsive hydrogels, we can make them with polymers which have both hydrophilic and hydrophobic parts. So as the temperature increases, if you follow the points written on the slide, as the temperature increases, the hydrophobic interactions will become more stronger and the hydrogen bonds become weaker. So once you put this hydrogel into water, it will definitely interact with water because of the hydrophilic parts. So as you increase the temperature, this interaction between the water and the hydrophilic part will break and the hydrophobic interactions become more dominant. So as a result of which the hydrogels will start shrinking due to this interaction, this hydrophobic interactions, and that's why at high temperatures they will start shrinking and they lose their solubility. So that's the right option here. Any doubts here? If not, we can move towards the next question. The next is what happens during phase 3 clinical trials, and we have to choose two correct options. Anybody wants to volunteer?
Option first, madam.
Okay, that is half of the answer. Do you have any other option? Since we are supposed to choose two.
Option 1 and 3.
Okay, yeah. So
1 and 3, ma'am.
yeah, both of you are correct. The right option is safe dosage is determined and it is generally a multi-layer and multi-centered process. So as I said, we have divided the clinical trials into three phases, where phase 3 is a multi-center involvement, meaning you do the same study at the same time at different centers just to cover a majority of the population, and also safe dosage is determined in the study. So the rest of the two options, the second option is the study is localized to very specific region and age group, which is actually a parameter of phase 2 clinical trials. And the last is studies are performed on large animals such as primates is also false because clinical trials are only performed on humans. Primates is one step before the clinical trials. So phase 3 includes safe dosage is determined and it's a multi-centered process. Clear? Okay, then let's look at the next question. Next is what is meant by sterilization? Improving the quality, improving the concentration of product, elimination of contamination, or purification of products. Any guesses?
Option 3, madam.
Okay,
4.
someone said option 3 and someone said option 4. Okay. Thanks for answering. Anybody else? No more answers? Okay, let's discuss the answer then. The correct option is elimination of contamination. So sterilization is nothing but a process where you try to eliminate any living form which might be present in your drug formulation. So this will eradicate all your viruses, all your uh, uh, bacteria or any spores of the bacteria, etc. So sterilization is essentially done to remove all any contamination which might be there. So the rest of the options are irrelevant because improving the quality is not what sterilization is. We are definitely not improving concentration of product, and we are also not purifying the product. So purification comes when you have some impurities which are other than living matter. So that is where your purification will come. Let's say you have developed a protein which is going to act as a drug, and you are isolating it from a bacterial. Let's say you have insulin and a particular bacteria produces your insulin, let's say E. coli, and you want to then isolate this insulin from the bacteria. So you'll start purifying it. You'll remove all the proteins, the DNA which might be present in your solution to get to the insulin, and then later on you'll sterilize it so that no other contamination is present. So sterilization is essentially elimination of contamination. I hope this is making sense. Any doubts here?
No, ma'am.
Yeah, thank you. Then let's look at the next question. The next is, which of the following are not examples of active targeting? And two correct options. Any volunteers?
Option 4 and 5.
Option 4 and 5 is EPR effect and direct local delivery to target organ. Okay. So essentially we are supposed to look at what can be the examples of passive targeting. Anybody else wants to take a guess? No? Okay, yeah, you were correct. The right answer is EPR effect and direct local delivery to target organ. So as I said, targeted drug delivery can be divided as passive targeting and active targeting. Passive targeting, where you take advantage of the environment that is, or you want to deliver drugs to a particular organ. So it can be EPR effect, it can be tumor environment. Tumor environment because if you've read about studies which look at the local environment in tumor, it is usually acidic. So you can formulate your drug in such a way that you encapsulate your drug in a polymer, and that polymer can be made pH responsive, meaning that it will start delivering the drug at pH which is lower than your physiological pH, which is found in tumor environment. And it can also be directed local delivery. Like I said, if you want to deliver something to your lungs, you can take the inhalation route. So that's passive targeting. Rest of the options are active targeting. So the question is asking us which are not examples of active targeting. The correct answer is EPR effect and direct local delivery to target organs. Okay, the next question. Next is, what are the main disadvantages of aptamer? And there are two correct options. Anyone wants
Option
to try?
2 and 4.
2 is they do not interact with negatively charged targets, and 4th is can be detected by TLRs. Okay, anyone else? Thanks for answering. No other responses? We can look at the answer then. So yeah, you were correct. The right option is they do not interact with negatively charged targets and can be detected by toll-like receptors. These are actually shortcomings of aptamers. So as I said, aptamers are nothing but nucleotide chains. So this is your DNA and RNA. So they are anyways negatively charged, because of the phosphate groups present in the nucleotides. So because of the negative charge, they don't interact with negatively charged targets. For example, let's say you're targeting your drug delivery to a particular cell and it has receptors on it, or it has so the lipid layer which is present in the membrane might be negatively charged. So if you're trying to deliver your drug into such a particular cell where the surface is negatively charged, then your aptamers might not have much interaction with these cells. So since they are negatively charged, so they'll start repelling each other. Another shortcoming is they can be detected by toll-like receptors as foreign DNA. So toll-like receptors are nothing but protein receptors which are present on your immune cells, such as macrophage cell lines or your macrophages or your dendritic cells. So these toll-like receptors are very efficient in detecting any foreign DNA, which is most relevant in case of microbial infection. So they are very good at detecting microbial DNA. So if you because your aptamer is now a synthetically produced DNA, they might elicit a immune response because they'll be considered foreign. So your toll-like receptors might detect them and then it might be degraded. So these are few of the disadvantages of aptamer system. Clear? Okay, I'll move on towards the next question. The next is, which of the following statements is false for enteric coatings? And there are a bunch of options. We have to select one right option, which is not a characteristic of enteric coatings. Any guesses?
Second option.
Second, it is insoluble at low pH and soluble at physiologic pH. Okay, anyone else?
Fourth one allows delivery to the stomach.
Okay, anyone else wants to try? Thanks for answering, both of you.
Option 3, ma'am.
Protects acid-sensitive drugs from gastric juices. Okay, so I hope you're reading the question correct. It's asking what is false for enteric coatings.
4, ma'am.
Yeah, okay, so a bunch of responses. I'll discuss the answer. So the correct answer, it allows delivery of drugs in stomach. So as I said, the question is asking us what is false about enteric coatings. So enteric coatings, as we have discussed a couple of times, these coatings are essentially made so that you prevent the drug dissolution in stomach. So if you see the figure here, it's coated with a polymer that contains carboxylic acids, and at lower pH these carboxylic acids will be protonated. That is why they'll have low solubility. They'll all be in the COOH form, and since these are acids, as the pH increases, which happens when the drug goes from stomach to small intestine, these carboxylic acids will be deprotonated at higher pH, meaning they'll lose their proton. They'll be CO minus and H plus. Now since these are ionic species, they'll have more solubility. The polymer membrane will start dissolving because of this, because of interaction with water, and then your drug can be easily released. So if you look at the options, first is pH responsive. Enteric coatings are pH responsive. We have to look for something that is false about enteric coatings. Second option was insoluble at low pH and soluble at physiologic pH, which is also right. At low pH, since it's protonated, COH, it will not dissolve. At higher pH, since it is CO minus, it will be more soluble. So that's also correct option if the question was true about enteric coatings. And then third is protects acid sensitive drugs from gastric juices. This is why we have made enteric coatings. We don't want the drug to be released in stomach. Fourth was allows delivery of drugs in the stomach, which is actually false, because enteric coatings will not allow anything to dissolve in stomach, so the drug will directly be released into your intestine. And then delivers drugs to small intestine, which is again correct. Can be made of polycarboxylic acids, again correct. So the right answer for this question is it allows delivery of drugs in the stomach. Is this clear? Because there was some confusion with second and third option.
Yes, ma'am.
Yeah, thank you. I'll move on towards the next question then. Next was poly N diethyl is widely used as a temperature responsive hydrogel because of its LCST in the range of. So this is a straightforward question. Might have been mentioned in the slides.
Option 1, ma'am.
Option 1. Okay, yeah. There's nothing much to discuss here. So as I said, this is one example of temperature responsive hydrogel, and it is widely used because it has its LCST in the range of 25 to 32 degrees Celsius. So this was mentioned in the slides. Let's look at the next question. So next was, which of the following functional groups would make coating non-liable to acid damage in stomach? We just discussed about it. Anybody wants to take a guess?
Option 3.
Option 3. Anyone else? Thanks for answering.
Yes, ma'am. Third option.
Yeah, okay. Yeah, both of you are correct. The right option is carboxylic acid group. If you read the question carefully, it is the answer which we are supposed to choose is a functional group which would make coating non-liable to acid damage in stomach, which means a functional group which will prevent acid damage in stomach. So that is nothing but your COH groups, as we have discussed multiple times in enteric coatings also. Your COH groups will be insoluble at the pH which is found in stomach, and it will start releasing the drug once it moves towards the intestine. Is this clear? If there are no doubts, I'll move on towards the last question then. So the last question is, what assays can be conducted to test for nanoparticle toxicity in vitro? And we have to choose two correct options. Anyone?
I said 2 and 4.
2 and 4. Okay, hemolysis assay and transfection. Okay, anyone else wants to try? Thanks for answering. We'll discuss the answer. No more responses. Okay, then I'll discuss the answer. The correct answer is hemolysis assay and cell metabolism assay. So hemolysis, as I said, so first of all, we need to test the toxicity of the nanoparticle which we have made. So there are various ways to do this and various different assays which we can use. First, one of it is hemolysis assay, where you want to see whether your nanoparticle is causing lysis of your blood cells. So you can take blood from, it can be from a human or it can be from an animal. It can be a mouse blood also, and you just incubate your nanoparticles with the blood, and you check for whether the cells are in the right morphology, if they are growing happily or if they're lysing. So this is hemolysis assay. Another example is cell metabolism assay, where you, let's say you take a fibroblast cell line or a macrophage cell line, and you put in your nanoparticles in the media where the cells are growing in vitro. Everything is done in vitro, and you check the metabolism of these cells, whether they are growing happily, whether they are under any stress, are they stressed, are they shrinking, how do they look. So all these assays can be done to test the toxicity of nanoparticle in vitro. First option, that is protein estimation, is nothing but quantifying protein, which is not actually going to tell you whether the nanoparticle is toxic or not. Similarly, transfection is nothing but the process where you infect a cell, mammalian cell with virus. So that process is also not going to tell you whether your nanoparticle is toxic or not. So the right option is hemolysis assay and cell metabolism assay. Is this clear?
Yes, ma'am.
Yeah, so we have discussed this assignment completely. If anybody wants me to repeat any of the questions, I can do so. So this was the last assignment, and I hope you guys have learned something from the 12 sessions which we had. Any doubts?
Ma'am, many negative markings are there.
Yeah, so negative marking will be there for multiple choice questions where you have to choose one or two options. So negative marking will only be not there for numericals, where you have to enter a numeric value You will be given, there'll be a blank box where you have to enter the value. So only those questions are exempted from negative marking. I think rest of the questions will have negative marking. You will get the instructions before the exam starts. In that it will be clearly mentioned. So please go through the instructions very carefully. You can spend some time reading the instructions first, and then you can go ahead with solving.
Thank you.
Yeah.
And regarding the assignments, minimum 8 assignments should be there, no, madam?
Sorry, you're not clear. Can you please repeat?
Assignments minimum, the marks will be taken for 8 assignments, no, ma'am?
Yes. Yeah. It will be considered some 8 or 9 assignments out of 10 for your grading, you're saying, right? For the final score.
Yes, ma'am.
Yeah.
Totally, how many questions will be there in the examination, ma'am?
I don't know about how many questions. The total marks will be 100 because it can be divided. So there'll be some questions will be single choice questions where only one option will be correct. Some questions will be multiple choice questions where 2 options might be correct, and some will be numericals. So I don't know how these will be distributed, but the final marks will be 100.
Okay. And how many hours the examination will be?
I'm not sure, it might be 2 or 3 hours. If you have doubts about the exam, I think you can post it in the forum. So the current TAs for the course can take care of these questions.
Yes,
I think
ma'am.
it's for 3 hours. Yeah, you can just confirm it with them.
Yes, thank you.
Yeah, thank you.
Thank
I lost you again. Can you repeat?
Thanks for having a good sessions for us, ma'am.
Sorry?
Thanks for having 12 week sessions for us.
Oh yeah, you're most welcome. I just tried explaining stuff to you.
Excuse madam.
Yeah, go ahead.
What videos now we are having in the mail regarding these 12 weeks lecture videos?
Okay.
Whenever we need to open those link, whether we will get the information, otherwise it is going to be disappeared.
No. So this will be on a YouTube channel. So that will be there forever. If you have access to these links, you can open it anytime you want. I don't think there'll be a time limit for that.
If any books is there to study for this subject, ma'am?
So I'm not sure. So I never refer to a book. So I've taken this course. So I have taken this course from the same professor in my institute as well. So I have not referred to any particular book. But if you go through the slides, there will be some references mentioned in the slides as well, where the professor has discussed some concepts from particular research papers. So the links are provided. So you can go through those links once.
Okay.
So is there any another channel to meet that Rachit Agrawal sir, madam?
Another YouTube channel, you mean?
Whatever it may be, either mail ID, whatever it may be. Actually, what are the thing he explained all this theoretical thing.
Right.
So you go for practical purpose, lot of problems will arise.
Right.
So we need to rectify those practical problems. If there is any separate mail ID, maybe is it available or not?
Oh, you want to contact Rajat?
Yes, madam.
Okay, so you can just type Rajat Agarwal IISc in Google. You will get his web page. So on that
Okay,
web page, you'll get his email ID. You can directly mail him saying that you want to discuss few of the concepts discussed in his course. So according to his availability, he'll respond to you.
Okay, what I need to give the madam?
Nothing. You can just mail the professor. You can search
Rachit
for
Agrawal IISc, madam.
Yeah. Search for Rajat Agarwal IISc in Google, and you'll get his web page.
Okay.
His website for his lab, and there you'll have his email ID.
Okay, madam. Thank you very much.
Yeah, most welcome.
Thank you, ma'am.
Thank you.
Thank you, ma'am.
Thanks.
Is there any theory question in exam, ma'am?
Theory question meaning you won't have to write long answers. You just have to choose options. That's all. You won't have any theoretical question where you have to answer in brief. All the questions will be multiple choice questions and numericals. In numerical questions, you just have to enter the answer. You don't have to describe anything.
Thank you.
Yeah, welcome. If there are no more doubts, I'll stop the meeting. anybody have any more questions?
Please explain first one, madam.
The first question of this session.
Yes, madam.
Yeah, just a minute. I'll flash the question first. Yeah, so the first question was what are GMP and QS regulations? GMP regulations apply to things which are intended to be used in humans. That was the correct answer. So GMP regulations are nothing but good manufacturing practice. So the regulatory bodies which are going to give you approval for your drug system need to look whether you have made the drug in clean conditions, in sterile conditions. So these regulations require that whatever manufacturing you have is of the utmost quality. Whenever you will be manufacturing it or packaging, labeling, storage, installation, everything should be in contamination free zones. So these are nothing but good manufacturing practices, and you need to produce your drug in these practices to get approval for something that is to be used in humans. So that was the question.
Okay, madam. Thank you.
Yeah, welcome. That's all. Shall I stop the meeting then? Amruta and Uzma, do you have any doubts? Okay, I'm assuming you don't have any more questions. Then I can stop the meeting here. Thanks for joining.